Thank you very much, Flavia, for the introduction. Good morning, everybody. It's a real pleasure to be here in Brazil, in Rio. It's not every day you get to give a presentation while looking at Christ the Redeemer, so it's quite an amazing experience to stand here today. And actually, I've been here for a week now with my young baby, a 9-month-old baby, and my wife, and we've had such a brilliant holiday. The Brazilian people have been fantastic, I have to say, and we look forward to coming back and seeing more of the country in the near future. But I'll be talking today about, as Flavia mentioned, about preterm infants. And we've heard a little bit about this already this morning, and I'll be picking up actually from where the previous speakers left off. Okay, so we have heard a lot about the importance of the gut microbiome development during infancy. This is true across the life course, but it's especially true during infancy, where over the first year of life, the microbiome is most developmental. And over this time, it also plays really critical roles in shaping our immune systems and many other factors as shown on this slide and also many not shown. But in the case of a preterm infant, which I will define today as those born less than 32 weeks of gestation, we know that they have a very unnatural beginning to their life. They spend the first weeks and months of their life in intensive care. They therefore have much more likely to be colonized by hospital-acquired bacteria, which are not always the most beneficial bacteria to have. They are more likely to have, as mentioned by Anna earlier, C-section delivery, more likely to receive antibiotics, less likely to receive adequate volumes of maternal breast milk. And all of these factors combined, as I mentioned, perturb the microbiome, but also play really important roles in the outcomes of those infants. My group particularly study an intestinal disease called necrotizing enterocolitis, or NEC. This is the leading cause of death in preterm infants, at least in the Western world. It's an exaggerated inflammatory disease at essence, so because of the immaturity or prematurity of the infant and their immune system and their sort of anatomically, they are at a much higher risk of developing this disease. And it's the typical inflammatory cascade which leads to eventually necrosis of the bowel, and in the worst cases, those infants would undergo surgery and have that portion of their bowel resected. There's a huge mortality risk associated with this disease, and even in surviving infants following surgery, they have lifelong morbidity as a result of that short bowel syndrome. So as mentioned beautifully earlier, we published this study a few years ago now, and really just stressing here that of all the factors we think we should care about when it comes to an infant, breast milk is the primary driver of that infant gut microbiome. Other factors are, of course, important, but to much lesser extent than breast milk. And once breast milk is no longer received in the infant diet, the infant's microbiome really rapidly changes to look much more like that of adults. So there's been a lot of work trying to unpick the so-called bioactive components in breast milk, and a lot of researchers really focused on these bioactive components called human milk oligosaccharides, or abbreviated to HMOs. There's been a lot of interest in HMOs in particular because they're so abundant. So HMOs are the third most abundant component in breast milk. And despite this, they actually have no nutritional benefit to the infant. So they reach the small and large intestines intact, where they provide substrates for the microbes, for primarily beneficial bacteria. So we know when an infant is being breastfed, they have an increase in bacteria like bifidobacterium. In part, this is why bifidobacterium are used in the majority of probiotics, particularly those targeted for use during infancy. But it's not just them providing the substrates and that these bacteria are able to grow, but rather what those bacteria produce when they are colonizing the infant intestine. And these, as we heard earlier by Francisco, include things like short-chain fatty acids and other important beneficial metabolites. Much less well studied, but just important to be aware of, is these sugars may also act as decoy receptors and may also directly have immunomodulatory roles, but those aspects are much less well understood. Okay, so we know, as mentioned again earlier, breast milk is critically important to infant health. It is the primary protective factor against necrotizing enterocolitis in preterm infants. And we now know as well from this morning's session that it contains viable bacteria, and it also contains this plethora of bioactive components. So we've been really interested in studying bioactive components and microbes in milk. We term it mother's own milk, or MOM. So we've looked at the microbes and bioactive components. And of course, what this all means to the infant is really the focus. And so the infant microbiome has really been the focus of my career to date. And much more recently, we are starting to ask the question, all of these factors combined, what does it actually mean for that infant? How does it impact their health? How does it modulate their barrier function and integrity? And I'll talk about that today. So this is sort of to give an overview of the different topics I'll cover over the remainder of my presentation. And just to mention, if you do have any questions, I think the QR code is available on your packs, and people online, please do reach out and ask questions. This talk is maybe a little bit different to the previous ones you've heard. I'm going to deep dive into some of our very recent findings, most of which is unpublished. So it's quite detailed science, but I really encourage you to ask questions so I can explain better where needed so that you thoroughly understand the concepts that I'll talk about. Okay, so we'll begin with mother's own milk microbes. So this is work that was led by a former PhD student, now postdoc with me, Andrea Massey. And in Newcastle, we've established what we call the Great North Neonatal Biobank. This resource contains over 100,000 samples from preterm infants, so a really fantastic resource for a researcher like myself to go and access these important samples from babies and from their mothers to learn much more about the biology underpinning health and disease. So in this case, despite a lot of work, we didn't find any difference between the number of bacteria in breast milk between necrotizing enterocolitis and matched control babies. We didn't find any difference between what we call the microbiome profile. So each dot on this plot here is a sample, a stool sample, a milk sample, and it represents the microbes in that sample. The closer together those dots or samples are, it means their microbes are more similar. And what we'd expect to see if there was a big difference between diseases, two very distinct clusters, and of course that's not what we see here. So no difference in the overall microbiome profiles either between neck and control infants, and no difference either in the relative abundance of any of the bacterial genera that we detected as well. It is worth noting that we were able to culture all of these bacteria, although what I'm presenting here is molecular data. That just means that those bacteria are definitely there and they're definitely alive. They're just not associated in any way with disease. And we also quantified this and again found no difference. So this was a lot of work. It's currently being submitted for publication. And I think the takeaway really for me is that whilst there's a lot of attention currently on spiking donor breast milk with maternal breast milk to sort of seed the microbes, this is a procedure that carries risk and cost. And based on this evidence, if you're trying to improve outcomes relating to neck, I would argue that it's probably not a procedure we want to be adopting widely. It's not to say it has other uses for other diseases, but when neck is the focus, the milk microbes are really not important. But the bioactive components definitely are. So I'll use human milk oligosaccharides here as an example. Again, these sugars in breast milk that are really abundant, primarily prebiotics. When we collaborated with Professor Lars Bode in UC San Diego a few years ago, and we looked at the sugars present in these milk samples being received by our preterm infants, as you can see, we found very clear difference between those who went on to develop disease. And in particular, this was driven by a single sugar, a single HMO called disilacto-N-tetrose, or DSLNT. And you can see here on the plot on your right that if you are a healthy baby or going to be a healthy baby, you're much more likely to have high levels of this sugar in the milk you're receiving. And in infants who develop neck, this sugar was low, and in particular, it was below this threshold of 241 nanomoles per mil. And this is a really important threshold I'll come back to in a couple of slides' time, but with this threshold in our cohort in Newcastle, we can predict necrotizing enterocolitis based on a single sugar in milk with incredible sensitivity and specificity. So we're doing a lot of work now to follow this up, but it does raise questions around the ability to start to think about supplementing this sugar and maybe other HMOs into our infant diet. And of course, this is a big direction of travel now for formula milk companies. And just as I said, I've got my baby here. I spend a lot of time trying to find baby food in Brazil, which I'll tell you is not very easy. But the shelves are stacked with these formula products which contain HMOs. So it's fantastic in a way to see the excitement in the scientific community finding its way into a product. But often these HMOs are not necessarily what we find to be the ones that are most protective. And so there's still a lot more work needed, I think, to really refine which sugars, if any, we start to supplement with. Okay, and so I mentioned that threshold of DSLNT is critically important, but what does that mean for the infant? What's the potential mechanism of why infants who have more or less of this are more or less likely to develop disease? So we can look at the infant microbiome longitudinally over time, and this is something that we do regularly. This is using metagenomic sequencing, the sort of state-of-the-art sequencing for the community, for the microbiome field. And effectively, I'll draw your attention here to what we call preterm gut community types 4 and 5, so those on the bottom. And you can see infants who receive a lot of that particular sugar in their milk, they quickly transition into a community in their gut microbiome, which is dominated by bifidobacterium. But we don't see the same for those infants who were below that DSMT threshold. So importantly, this is still an association. This is not causality. This is not a mechanism. But we are able to link lack of a particular sugar in milk with infant microbiome development, and then ultimately the risk of that infant going on to develop this deadly disease in necrotizing enterocolitis. So probiotics has been one of the topics of questioning, which has appeared after all the talks this morning, and I think is obviously a really big, important topic in neonatology at the moment. We started using probiotics in Newcastle over a decade ago, and this in part was related to some of our early work that showed bifidobacterium, when it was high, those infants never went on to develop NEC or indeed sepsis. So we used Infloran and then Labinic. Infloran and Labinic contain the same two species, but importantly different strains of B. bifidum and Lactobacillus acidophilus, and Labinic additionally Bifidobacterium infantis. So in work led by Lauren Beck, again, former PhD, now postdoc with me, she profiled a huge number of infants longitudinally, and these are only healthy infants, what I'll show now. And unlike in term babies, where I talk about delivery mode being an important variable and maternal milk being the most important driver, we don't really see the same impact on preterm infants. Again, consider that these infants are in hospital, they're receiving a lot of antibiotics, and they receive universal antibiotics following birth. The factors associated with the microbiome development in preterm infants are very different to term infants. But probiotics, as you can see here, were really significant across all of the time points we measured, basically from the point probiotics were given on day 8 on average. You see it has a significant impact on how that infant's microbiome develops. And that, I think, would be expected to anybody. We are adding bacteria to the diet. We are adding DNA from those microbes to the infant intestine deliberately, so we are more than likely to detect those DNA coming out in the infant stool samples. There's a lot more work gone into this paper I won't have time to talk about today. Strain variation, persistence, and many other things around the ecology of the microbiome. If you're interested, it's all open access, and definitely encourage you to read it. But I really want to focus on our unpublished data, and one thing we found is, despite assuming that probiotics are going to have these impacts on the infant microbiome, what we see in the case of infants who go on to develop necrotizing enterocolitis is actually probiotics have no impact in that population. So this might explain why infants who do receive probiotics can still go on to develop disease. It is simply that they are not responding to that therapy. The use of probiotics, for whatever reason, we are still actively working on this, the probiotics are not having any impact in those babies who go on to develop disease. It begs a question whether we should be moving towards more of a personalized or precision probiotic therapy. Maybe we were simply giving these babies the wrong probiotics for them. And this is, again, something that we're really keen to understand better. Okay, so now in the last sort of part of my talk, I really want to move into some very new stuff that we've been working on. And this centers around this concept that breast milk contains bioactives. Breast milk is an incredibly complex biofluid, but it's been shaped over the course of evolution, which is not a wasteful process. And therefore, one assumes if something in breast milk is abundant and particular microbes are able to use that component, those microbes themselves are also likely to be beneficial. So we've done a lot of sort of screening and surveying to find what bacteria are able to use breast milk bioactive components, what do they produce when they do that, and how does that actually impact the infant, which is, of course, what we really care about. So a really simple experiment we can do in the lab is simply culture bacteria from preterm infant stool samples or breast milk samples or tissue samples, and then see if those bacteria can grow in the presence of different bioactives. And in this case, I'll talk about human milk oligosaccharides, but again, it's one of hundreds of different bioactive components to think about. So this is quite a complex heat map. I'll sort of talk you through it in 3 parts. What we're looking at here is which bacteria are able to or not to use different HMOs, which are listed across the top, these different sugars in milk. You can see this population, this cluster here of what we consider to be non-beneficial bacteria in general. We often call them pathobionts, and they don't use any of the HMOs, and in most cases, and in some cases, don't even use lactose. We have at the bottom here, on its sort of own branch of this cluster, Bifidobacterium bifidum, which appears to grow really well on HMOs. I would say this is a slight artifact of normalization to glucose. The reality is this particular bacteria doesn't grow well at all on anything, but it just grows better on sugars, on milk sugars than it does on glucose. But the real novel discovery here is that we have this huge range of different Clostridium isolates and species that are able to use human milk oligosaccharides, and this was previously unknown. Most people really assumed it was only the bifidobacterium in preterm infants that were consuming human milk oligosaccharides, but actually we find this to be the case also with many different Clostridiums. And you can see a lot of strain-to-strain variation in their ability to use HMOs, but in particular we focused on this isolate, AM1, because it can use DSLNT, that really magical HMO I mentioned earlier that we think is protective against NEC, probably through modulation of microbiome, and therefore we really want to focus on the bacteria that can use it. It also clustered most closely to our only probiotic here, which is that B. infantis. So many will ask the question, and rightly so, is Clostridium perfringens in particular not a pathogen? So I was involved in work led by Lindsay Hall's lab a couple of years ago and published last year, which really, in a nutshell, found that the vast majority of Clostridium perfringens in preterm infants is in healthy babies. They did, in some cases, culture this bug from NEC babies, but you can also culture bifidobacterium from these babies. So just because you can culture a bacteria doesn't mean that that is causing the disease. And really, the take-home from this paper was that there was a cluster you can see here on the bottom in dark blue, navy blue. This is a hypovirulent lineage, so this basically means that bacteria in this cluster were always from healthy babies and therefore generally may be beneficial. And then other work really has pointed towards Clostridium either doing very little in terms of causing NEC or actually probably being beneficial. In our own analysis, we find Clostridium perfringens to be higher in infants who are healthier and lowest in infants who develop NEC. So again, some really nice evidence that this bacteria that can use HMOs is actually probably beneficial. But why is it beneficial? So I mentioned earlier one of the critical part of the pathway is these bacteria colonize the infant intestine, but it's really what they produce that's important. That's what's having the impact on the host in large. So we know bifidobacterium in a head-to-head with Clostridium will produce more acetate. This is a particular short-chain fatty acid bifidobacterium are famous for producing. But our Clostridium isolates, when growing on these breast milk sugars, can produce a huge range of different short-chain fatty acids, in particular things like butyrate, which if you've ever followed this literature, you'll know is probably the most important short-chain fatty acid when it comes to not only infant health, but also health across the life course. The other metabolites that we've really digged into is what we call the tryptophan catabolites, and again, you can see a consistent pattern here that those beneficial metabolites are generally being produced by our Clostridium, in particular our Clostridium perfringens, and in many cases a bacteria called Clostridium tertium, compared to bifidobacterium. So here we have a bacteria not previously known to be probiotic, still hesitate to call it a probiotic, but it definitely is producing beneficial metabolites for the infant to use. So then the final part of this jigsaw is, what are these metabolites actually then doing to the infant? Sure, we think they're beneficial, but can we actually prove that they are? So what we do to get more at the mechanism in our cohorts is use what we call organoids. These are basically our ability to take resected tissue from preterm infants and regrow that baby's intestine in the lab. And we do this all the time whenever an infant has surgery in Newcastle. And we also have a co-culture model that allows us to actually then test bacteria in direct presence with those human mini guts, because we can control the oxygen across the epithelium. So what does the data look like? We spent a lot of time developing a model. We're spiking LPS, which is a cell wall component of microbes, as well as flagella, again, a microbial component. And this is really our inflammatory stimuli. This causes our mini guts, our preterm epithelium, to become inflamed. And then what we can do is add our postbiotics, so we can grow Clostridium on sugars, and we can remove all of the clostridial cells, so all that's left in that supernatant is just the beneficial, well, the metabolites that we presume to be beneficial. And that's what we're seeing here. When we look at the epithelial layer, we see that these Clostridium postbiotics are able to increase barrier integrity, which would be a critical, important factor for infants with disease. And we can look at actually the pro-inflammatory cytokine release as well. So I've used IL-8 here as an example, but this applies to all of the cytokines we've measured. And you can see when we provide the stimuli, this is on the far right of the plot, as expected, the cells are very inflamed. They produce a lot of IL-8. When we add our postbiotic without any stimuli, you can see that next to the control, it doesn't itself induce any inflammation. But most importantly, when we add our Clostridium postbiotic, we are actually able to reduce that inflammatory stimuli, both apically and also basolaterally. So again, really potentially important for an infant who has a disease like necrotizing enterocolitis, a pro-inflammatory disease. The ability of these postbiotics to reduce that inflammatory insult could be life-saving in that population. And in my final word of note, and we can maybe pick up on this more in the discussion, but there's been a huge debate recently around the use of live microbial therapy in preterm infants, particularly in the US, due to a blood culture positive and ultimately fatality due to a probiotic species. Moving away from live therapy into this element of postbiotics may have a much safer and potentially equal efficacy for infants. Okay, so I'll leave it there. I will thank the many people who's been involved in the work. Hopefully I've given them a nod as I've moved through the presentation, as well also to the funders. Again, a big thank you to you all for your attention, and I'm happy to take any questions you might have.
Thank you, Chris, for your wonderful presentation. Well, we got so many messages here again. Well, everyone is very happy and, well, praising you for your great presentation.
Thank you.
I have a question here. From what we get from your presentation regarding the prevention of NEC, necrotizing enterocolitis, is it right to say that rather than looking for a proper probiotic in order to prevent NEC, we should look for the proper food to feed the right bacteria?
Yeah, I think that's a very valid point. We know that if an infant is typically breastfed, that is a natural way of increasing bifidobacterium, and perhaps even circumvents the need to pay for probiotic therapy and to use probiotic therapy. And as a general rule, not just for NEC, but as a way to live, the more natural something is often better. So yeah, artificially adding bacteria it's one mechanism, but if we can do that naturally, of course, that's always better. The question for a preterm infant in a neonatal intensive care unit, though, is that bifidobacterium has to come from somewhere. We know that one source of transmission is through fecal oral transmission during vaginal delivery in a term baby. But as mentioned, preterm infants are much more likely to be delivered by cesarean section and therefore potentially miss that critical exposure during delivery from the maternal feces. And therefore, in those cases, we might need some probiotic therapy just to get some bifidobacterium into these babies. And there you would hope that you can modulate it with diet rather than continually providing the drug or the probiotic.
Right, thank you. There's another question here. Is there a difference of using HMOs from human milk or comparing to HMOs in milk formulas?
So human milk oligosaccharides, by definition, are isolated from human milk, but structurally will be similar or identical in many cases to what you can synthesize or extract from non-human mammalian milk. But it's worth noting that in human milk, they are, as I say, the third most abundant component, more abundant than things like protein, which is very different to what you would naturally find, for instance, in bovine milk. Human milk oligosaccharides are near enough absent from bovine milk. So it's often the case that they need to be synthesized biochemically or using microbes to make these sugars, rather than extracting them from non-human species. But yeah, so companies will call them human milk oligosaccharides. Some will call them human-like milk oligosaccharides, so not quite exactly human, but structurally they're very similar or the same, and functionally they will behave the same.
Right, thank you. And apart from this DSLNT for the prevention of NEC, any other HMO would you identify as important?
So this is a good question. The short answer is no, really. It's in our studies and in other studies available today, DSLNT has really stood out, actually. So what I showed from our Newcastle cohort actually validated work that Lars had performed earlier very nicely. His cohort was slightly smaller, but it was a generally Hispanic, very different sort of ethnicity makeup to our Newcastle cohort, which is largely sort of white, Caucasian. But we've seen very consistent results. So in a way, that's very promising. What we're trying to do now is use different analytical methods just to confirm that that's definitely the case when we use other instruments to measure these sugars, and then ultimately progress this much more towards a biomarker. It's a good question as well, because DSLNT is, compared to some of the other HMOs, and particularly the HMOs you'll find in current formula milk products, is relatively complex, and therefore much more expensive as it stands right now to produce, which is one of the reasons that you won't find clinical trials using this particular sugar, because it's just not as available as some of the more common sugars like 2FL, which you will see in studies and in human milk products.
Right.
Bovine milk products.
Flavia, do you want to make any question or comment?
I want the space for the question.
Right. Anyone from the audience? Yes, do we have a microphone for, there's a question from the audience. While we wait for the microphone to arrive to you, there's a question. I'll read it as it came to me. Previously, individual microbiomes was compared to a fingerprint. Is there a critical window for establishing a microbiome for infant programming?
Yeah, I mean, it's a very good question. I'm sure there is. We don't know what it is, and I don't think it's a binary cutoff. It's a very dynamic process during early life. So again, picking up on some of the comments from earlier, there's many reasons why a mother can't or won't be able to breastfeed their infant, but there is an argument that even some breast milk really early can have really important impacts. And so there is some epidemiological evidence that the longer the better, particularly up to 1 year of life. That hasn't necessarily been linked well to microbiome, but it has been postulated as one of the primary mechanisms whereby prolonged breastfeeding is associated with better outcomes because it changes the microbiome over a longer period of time. But yeah, able to put an exact sort of number of weeks or months or years we just don't have that current data. And it probably varies from individual to individual as well.
Right. We have a question from the audience. If we have questions here, can you just say your name, where do you come from, please?
Good morning, excellent talk. My name is Chahar Belkuri. I'm a pediatric infectious disease and neonatologist, coordinator of neonatal intensive care unit in Brazil. And so my question is, I think that probiotics in Brazil, it's a controversial issue. So what's your current opinion? In my experience, it's not a good experience in using probiotics in Brazil. And so what's your advice to use in Brazil? Because my experience is not a good experience, but I have no experience in Brazil to share.
Yeah.
So please, thank you very much.
Can I maybe just ask you to elaborate?
Congratulations for this study.
Yeah, thank you. Just quickly before you hand the microphone back, when you say you have a bad experience, are you able to maybe just elaborate very quickly on
yes,
what that is?
Yes, I have used it 1 or 2 times, and so it's not good to use it because that time the baby just had a bad outcome, and
okay.
I didn't like it.
Okay.
So I had to make some interventions, and it's not so good to the
Okay. So thank you very much for the question, and it's a really important point. I unfortunately will not advise one way or another because I'm not medically trained and I have no medical background, but I can certainly give my opinion on this. I think in the UK now we have probiotic use in around 40% of our NICUs. It's a very cheap therapy. It's generally regarded as safe, but of course, as the FDA rightly pointed out recently, there are cases where infants will develop sepsis and ultimately fatal sepsis as a result of probiotic administration. So there is always something to consider when thinking about using probiotics in a unit like a neonatal intensive care unit when the risk of sepsis is extremely high. My argument to someone like the FDA would be, or an organization like the FDA, would be that those infants have very leaky guts, and that infant may well have developed sepsis of an E. coli or a staphylococcus or something else that was dominant. It just so happened that the probiotic isolate in that particular case translocated. It doesn't make it a pathogen, it just meant the baby was very vulnerable. But
just to say, he's asking about colostrum therapy.
So all of our infants, or the vast majority of our infants, will receive maternal colostrum, and our breast milk receipt rate is very good in Newcastle, and actually our NEC rate at the moment is very low, and we think that's a combination of maternal own milk has gone up because of better training, nurses, lactation consultants, and all of these fabulous things. It's probably because we use probiotics, and it's probably because we also use donor milk. It's hard to say one way or another what is really causing it, but these factors combined seem to really help. Of course, we still have cases of NEC. We have 22-week babies being delivered. The risk is very high. We will never eradicate NEC, but we can hopefully reduce the risk.
Yeah. Let me add just a comment about the use of probiotic in the prevention of necrotizing enterocolitis, because I guess this was your focus, right? So there is ASPGAN guidelines identifying the strain. And also Anya wants to give there is this, and maybe, following the specific strain. And let me add you something that also Chris say. When you use a probiotic in a newborn, it's for prevention of necrotizing enterocolitis, not for treatment. So the problem is when the baby has to start, not when the baby has got already the necrotizing enterocolitis. Because the true risk for having probiotics is when the newborn is unstable. What means unstable? Low blood pH, heart rate, low heart rate, blood pressure. So the vital signs unstable. Do not think to cure necrotizing enterocolitis with probiotic, but just prevent. So when the baby is born. So Anya wants to add something. Yeah. You will and Anya Sayeska will have a talk later in this morning. But this is the core. Not to use in treatment, but in prevention. If you go in prevention, you're out.
And maybe just sort of a final point on that. As a clinician prescribing the probiotics, the meta-analysis do overall support that they will reduce the risk of NEC, but it's not possible to say this baby and this baby will have got NEC if I hadn't used probiotics. You'll never know the babies you've saved, but you'll always know the baby that goes on to develop a probiotic sepsis as a result. So I can understand clinically the burden or responsibility is massive. It's easy for me to sit here and say meta-analyses say you should use them and the FDA is completely wrong. But as a clinician, that you made that conscious decision to give that baby that bacteria, now they've potentially died because of infection of that bacteria. So you'll only know about the babies that ultimately that happens to, and never the babies you've saved. You'll just have to entrust in yourself that you have saved more babies than any fatality as a result of that intervention. And the final point, which I think is often overlooked, and maybe in your case is a bit different because it sounds like you prescribed it to one individual, but in our unit in Newcastle, we use it universally. It's not something parents can consent to. If they're in Newcastle and their baby's premature, they are going to get the probiotic that's in use at that time. And in many ways, it would be disingenuous to suggest that they have a decision to make, because once a probiotic is used in a neonatal intensive care unit, it will spread across the unit. So whether you just want to give a baby a probiotic or not, if the baby next to that baby is getting a probiotic, it's going to be cross-colonizing into other babies. So we use it universally as a standard of care. And I think it's the only way to do it truthfully. If you consent parents, give them the belief that they've got a decision to make, the reality is they really don't. But it's a very good question, and thank you for bringing that up.
Excellent, Chris. Well, we have so many questions here. I want just to tell you there's some questions that were already discussed in previous presentations. So if you want to return to the other presentations when watching them, it will be fine. There's a question here on short-chain fatty acids, particularly butyrate. Yeah. So if butyrate is so important, why don't we use...
O que é o butyrate? Por que nós não usamos isso para prevenção e tratamento? Esse é um excelente ponto. Nós temos alguns estudos diretamente administrados.
Animals, and yeah, there has been improved outcomes. There probably is a Goldilocks zone of butyrate, so there's been some evidence, albeit in terrible models, that too much butyrate can be then toxic or pro-inflammatory. So like most things, too much of anything can also be a bad thing. But yeah, it's very possible that we could give butyrate or other short-chain fatty acid supplement. The argument I would make with a more complex thing like a postbiotic is it's not just, well, we really don't know what the mediator of improved outcomes is. It probably varies from individual to individual. So by having a much more complex postbiotic therapy, you're potentially covering many more bases. In the same way that a fecal microbiome transplant works for something like C. diff, we really don't know what it is about that complex therapy that actually is the effector, but we know it works. And the postbiotic for me would be the safest way to give a really broad therapy. But the question is exactly right in theory.
Right, thank you. There's another interesting question regarding nutritional status of the mother and the composition of HMO. Are you aware of any studies on nutritional status of the mother and the levels of DSLNT in breast milk?
So that is a fantastic question, and unfortunately in Newcastle we are very focused on the infants. We're neonatologists. When the infant comes into the world, we care at that point. And from a researcher perspective, unfortunately, we haven't then got good information on maternal diet and frankly, other aspects around the maternal health. So I can't say with certainty from our cohorts what aspects of maternal diet might change HMO composition and in particular increase DSLNT. But that's a type of study that I would really love to do. There's very little evidence. We know that diet can impact HMO composition, but we don't know specifically what components of a maternal diet can increase certain HMOs. Those questions will be addressed in the years to come. We're working on it, but unfortunately we did not have that data for our historical cohorts.
Right. And does this apply also to the content of HMOs in breast milk and nutritional status of the baby and growth?
So we'll know the nutritional status of the baby, and many others do as well, but and again, when it comes to DSLNT specifically, there hasn't been any big studies. We lack the data to be able to say definitively what. It might not even be maternal diet related. It could be ethnicity or genetic or anything, but we really lack the information to be able to say with confidence what one can do to increase DSLNT in their milk.
Right, right. There's another question on, the effect of antibiotics, positive or negative causative effect in necrotizing enterocolitis in preterm babies.
Yeah, I mean, it's the frontline therapy for infants in our cohorts with NEC. It's pretty standardized in the UK, and I'm sure elsewhere in the rest of the world that they'll have 5 days of metronidazole. And often we can medically manage NEC with cessation of feed, so we reduce the sort of insult to the gut. Of course, that has connotations for the infant because they go nil by mouth for periods of time and maybe use parental nutrition. And with the antibiotic therapy, in many cases, that's enough to get the infant over that bout and they don't require surgery. So I do think antibiotics, particularly metronidazole, are critically important for the treatment of NEC. As Anna mentioned earlier, antibiotic use should be stewarded. It shouldn't be something that we just throw around, but it's still a really important therapy that we should be using, and certainly is in the case of NEC. The long-term impacts of that are very difficult to study because all infants in our NICU and in NICUs around the world often have antibiotics. and then disentangling what the long-term impact of that is very challenging.
All right, thank you, Chris. There's an interesting question regarding Clostridioides. The question is, Clostridioides can be considered as probiotics, the question, and what complications with these agents you had in your studies, considering it can, Clostridioides can actually cause diseases?
So, like most things, it's nuanced. There are pathogenic forms of this bacteria. I mean, you can look at E. coli. There's probiotic E. coli and there's pathogenic E. coli, and we see very similar things with Clostridium and Clostridium perfringens. So we focused our work on what we believe to be the beneficial Clostridium perfringens, those that lack particular toxin genes that would be potentially detrimental to the host. We haven't done any work with those particular isolates. And one thing I will say about the sort of probiotic use of a bacteria like Clostridium over something like Bifidobacterium, which is definitely going to be considered more widespread as safe, is really when you think about ecology and you think about competitive exclusion, if you really want a good bacteria to outcompete a bad bacteria, that good bacteria needs to be as similar as possible to the bad thing it's trying to outcompete, because it will occupy the same niche, it will consume the same substrates. And so Bifidobacterium is going to be a poor excluder of pathogenic Clostridium, but beneficial Clostridium will be a very good excluder of pathogenic Clostridium, because it will sit in the niches in the gut and prevent those pathogenic Clostridium from binding. So this is the argument I'm putting forward. That's very hard to prove mechanistically and experimentally, but from an ecology framework, I believe that, to me at least, that makes sense.
Thank you, Chris. Well, again, time just flew. In this discussion, I thank you, Chris, for your great presentation,
Thank you very
and
much.
everyone for the questions. Thank you.